Hello everyone, I am Rishabh Dev Singh. I am a graduate student in Professor Kyungyo Kim's lab. I'll be presenting our research on the generation of adult pluripotent cells for therapeutic applications. So before I discuss about adult pluripotent cells, I would like to discuss about induced pluripotent stem cells and their application. So iPSCs are pluripotent cells which can be differentiated to multiple lineages and they have plasticity similar to embryonic stem cells. And this was first reported by Professor Yamanaka using a set of four transcription factors, commonly known as Yamanaka factors, for which Professor Yamanaka got the Nobel Prize in 2012. So these iPSCs can be differentiated to different cell types, and these cell types can be used for multiple applications like disease modeling, drug screening, cell-based therapy for regenerative medicine. So despite the tremendous potential, these iPSCs have multiple limitations like genomic instability, tumorigenic potential. So one of the main challenges for the bench-to-bed translation of iPSC-based therapy is the tumorigenic potential of iPSCs. So it has been reported in multiple animal models that if we transplant iPSCs within the body, it forms tumor, and that is one of the main reasons why iPSC based derived cells need to be highly pure to make sure that we don't have iPSC contamination before transplantation. Other limitations include genome instability, which is mediated by high DNA damage. Then we have low efficiency, which is as low as 0.1%. Then time taking, the process of iPSC generation and their further differentiation to a subsequent lineage is a very time consuming process. So these are the limitations which limit the application of or the translation of iPSCs from bench to bed, highlighting the need for an alternative cell source. So in our research, we are generating adult pluripotent cells which are alternative to iPSCs. So we are using fibroblast cells, and within 2 days of small molecules treatment, we are generating iPSCs. Adult pluripotent cells. And we believe that these adult pluripotent cells can bypass the limitations of iPSCs. We will further discuss this in the subsequent slides. So adult pluripotent cells can be differentiated to all the cells from different germ layers and cells associated with multiple organs, and these cells can be used for cell-based therapy, and that thus these adult pluripotent cells can be used as an alternative to iPSCs. So in the subsequent slides, I will be discussing the results and highlighting how the APCs can be used to generate different cell types, and it can be used as an alternative to iPSCs. So in the first slide, we discuss about the generation of neural stem cells and neural crest cells, which are the main cells of central nervous system. So here we see that fibroblasts can be generated or differentiated to adult pluripotent cells using a cocktail of small molecules within just 2 days of treatment. And these adult pluripotent cells can be further differentiated to form neural stem cells and neural crest cells. And these neural stem cells and crest cells can be differentiated to several types of cells which are found in the central nervous system, like neurons, astrocyte, glial cells, Schwann cells, and mesenchymal stem cells. And these cells can have application in multiple diseases or degenerative diseases like brain stroke, spinal cord injury. So when there is such a tissue abnormality, these cells can be differentiated or generated from APCs and can be transplanted for therapeutic purposes. So one of the main advantages of these cells is they are patient derived cells, and they will not have any immune rejection. So it is a kind of precision medicine where we are using just patient derived cells for therapeutic applications. So in this slide, we show the generation of neural stem cells, neural crest cells, or the cells from ectodermal lineage. Next, we see the generation of cells from mesodermal lineages from adult pluripotent cells. So we see we have results on the generation of smooth muscle cells, and we have results on the generation of hematopoietic stem cells. So smooth muscle cells can be used for muscle degenerative diseases and can be used for generating muscles. So when there is any disease, degenerative muscle related degenerative diseases, these cells can just be transplanted in the patient and can help in the regeneration. We have hematopoietic stem cells generated with an efficiency of 34%, and this efficiency is very much similar to iPSC based protocols. So hematopoietic stem cells can be further differentiated to multiple types of blood cells or immune cells, and their application include one of the classical example for the immune cell application is cancer immunotherapy, which is gathering lots of attention nowadays. So these hematopoietic stem cells can be used for cancer immunotherapy, blood replacement therapy. So again, we have a proof that our APCs can be differentiated to form cells from the mesodermal lineage. Again, that's another proof for the pluripotency or ability of these cells to differentiate to different cell types. Then we have the results from the endodermal lineage cells, where we have generated pancreatic beta cells and hepatic progenitors from adult pluripotent cells. So we know that pancreatic beta cells are responsible for the release of insulin in the body, which is in basically in response to the glucose level. And when the pancreas cannot release the insulin, the glucose level increases in the bloodstream, and what we know is the result as diabetes. So one of the potential applications of the generated pancreatic beta cells is it can be used as a patient-based cell-based therapy, where we can transplant pancreatic beta cells generated from patient's own fibroblast cells, and these once these pancreatic beta cells are transplanted, they will release insulin in response to glucose, and that will maintain the glucose level in the bloodstream. The other application is the hepatic progenitor cells, which can be differentiated to generate hepatocytes, and these hepatocytes can be used for liver diseases like fibrosis, cirrhosis, or late stage fatty liver late stage cases. So in these three slides, we have discussed about the possibility or the ability of adult pluripotent cells to to differentiate into cells from the three germ layers, which is the main characteristic of iPSCs. So we have enough evidence to prove that our cells which we have generated can be served as an alternative to iPSCs. So here's a comparison between iPSCs and APCs. So we know that both these cells have pluripotent ability. They can be differentiated to multiple lineages of to cells from different organs, and they both have good potential for therapeutic application. But one of the main advantages of APCs over iPSCs is they do not form tumor. So we discussed that iPSCs have tumorigenic potential, but APCs do not. The other advantage is when we are generating APCs, there is minimal genomic instability or DNA damage induced, because we are using just a cocktail of small molecule for 2 days, and these 2 days of treatment is enough to induce APCs without significant DNA damage. And one of the main causes for DNA damage in iPSCs is the use of viral particles for the delivery of Yamanaka factors, but in our case we use just small molecules. And we have the time required for iPSCs. It takes close from 20 to 50 days depending on the protocol, but APCs can be generated within 2 days. So in summary, we have generated adult pluripotent cells from easily available fibroblasts, and these APCs can be generated within 2 days using a cocktail of small molecules. And these APCs can be further differentiated to multiple therapeutically active cells and potentially acting as an alternative to iPSCs, and it bypasses the major bottlenecks of iPSCs. So we can say that we have an alternative to iPSCs which can be used for cell-based therapy for regenerative medicine. Thank you.